cyclic and non-cyclic photophosphorylation. As we are discussing about photosynthesis and their process, in which there are two types of processes which are taking place in photosynthesis. One is light reaction, that is light dependent reaction or light reaction. We can also call it as photophosphorylation. And the another one is dark reaction. Okay, so far we are discussing about light reaction. Light reaction includes absorption of light, splitting of water, release of oxygen, and ATP and NADPH are also formed. So we are discussing all these. Now in cyclic and non-cyclic photophosphorylation, we are going to see how this ATP and NADPH are formed in this topic. First of all, let's learn what is photophosphorylation. Photo means light and phosphorylation means the ADP that is converted into ATP. That means one phosphate is being added. This process we say as phosphorylation. So altogether, photo, in the presence of light, this ADP is converting into ATP. So we say it as photophosphorylation. And now, as we already said, there are, in light reaction, it takes place in two cycles: cyclic photophosphorylation and non-cyclic photophosphorylation. Let's see about non-cyclic photophosphorylation. The non-cyclic photophosphorylation starts with photosystem 2. We have already discussed there are two types of photosystems. One is photosystem 1 and photosystem 2. So this non-cyclic photophosphorylation starts with photosystem 2, which has a light wavelength of which observes wavelength at 680 nanometers. So the chlorophyll A pigment along with its accessory pigments absorbs the light energy and in this chlorophyll A, the electrons, a pair of electrons, gets excited and they jump into the primary electron acceptor. Okay, from there, from primary electron acceptor, it moves down from higher level to the lower level, moves down to 3 electron acceptors. They are, one is plastoquinone, the second one is cytochrome complex, and the third one is plastocyanin. So the electron that is excited from primary electron acceptor moves to the plastoquinone first, and then it passes to cytochrome complex. Here in cytochrome complex, this electron uses energy where this ADP is converted to ATP, where in cytochrome complex. So after this, again this pair of electrons passes through plastocyanin. So near cytochrome, the energy is utilized to convert ADP to ATP synthesis. Okay, so that is why we are saying it as phosphorylation. So producing ATP. So in the presence of light it is taking place. That's why we call it as photophosphorylation. Okay, so in photosynthesis we have told that converting the light energy into chemical energy. That means light, the chlorophyll is absorbing light which is converted into chemical energy, that is ATP. Now the electron which is passed through plastocyanin is then shifted to photosystem 1. So photosystem 1 has a wavelength of P700. So in this photosystem 1 also, the chlorophyll A with its accessory pigments absorbs light, and a pair of electrons gets excited and they get shifted to primary electron acceptor. From there again, from higher level down, it passes through an electron acceptor, that is ferredoxin. Okay, from this ferredoxin then again it transfers to NADP plus. In photosystem 2 we got ATP. Now we need to get NADPH. So how we are going to get these protons? Let's see. In the splitting of water by photolysis. Photolysis is splitting of water. The water absorbs light and then it is break down into electrons, two protons and an oxygen as byproduct. Okay, see, this pair of electrons are added to photosystem 2 where the electrons are continuously lost. See, from photosystem 2 electrons are lost and they are get excited to electron acceptors. So these electrons that are lost in photosystem 2 must be replaced. So the photolysis of water, a pair of electrons are being added to photosystem 2. So it is replaced by water, photolysis of water. And now a pair of protons, oxygen is released as a byproduct and the remaining thing is a pair of protons. This proton is being added to NADP plus, which this is reduced to NADPH plus H plus, or we can write it as NADPH2. So we got ATP and NADPH. So why we are saying this as non-cyclic photophosphorylation is, photophosphorylation we already know because this phosphate is ADP is converted into ATP in the presence of light. We said it as photophosphorylation. Why we are calling it as non-cyclic? Because in chlorophyll the electrons that are lost goes to primary acceptor, then through 3 electron acceptors, and from there it is passing through photosystem 1, and from there again to primary acceptor, electron acceptor, and from there to ferredoxin as an electron acceptor and NADP. And the electron that is lost in photosystem 2 from chlorophyll A never return back again to chlorophyll A. So that is the reason we are telling it as non-cyclic. The lost electrons, pair of electrons from the chlorophyll A never again return back to chlorophyll A. Okay, so that is why we call it as non-cyclic photophosphorylation. The other one is cyclic photophosphorylation. So where does this non-cyclic photophosphorylation takes place is, it is in the thylakoids. And to be very specific, this photolysis of water takes place in the inner membrane of thylakoids. So this non-cyclic photophosphorylation, because it have pigments for the photosynthesis, it takes place in thylakoids. So this is about non-cyclic photophosphorylation, which is taking place in thylakoids. Now let's learn about cyclic photophosphorylation. So in cyclic photophosphorylation, it takes place in photosystem 1, that is 700 nanometers. Okay, it starts with photosystem 1, but in non-cyclic photophosphorylation, it starts with photosystem 2. It is at 680 nanometers. But in cyclic photophosphorylation, it starts at photosystem 1. Here in chlorophyll A, along with the accessory pigments, it absorbs sunlight. Here also a pair of electrons gets excited and reaches the primary electron acceptor. From the primary electron acceptor, the pair of electron moves to the electron acceptors, that is ferredoxin. From ferredoxin, it passes through plastoquinone, and from plastoquinone, it then passes to cytochrome complex, and from cytochrome complex, it passes to plastocyanin. So from plastocyanin, this pair of electrons moves to chlorophyll A. The electron, when it is passing from ferredoxin to plastoquinone, the ADP with inorganic phosphate helps in formation of ATP synthesis. And again, when this pair of electron is passed from plastoquinone to cytochrome complex, again the ADP with inorganic phosphate forms ATP synthesis. So only ATP is formed in this process. So as the result, from the electron, a pair of electron, as it is passing from chlorophyll A, then to primary electron acceptor, then it passes through ferredoxin, plastoquinone, cytochrome complex, and plastocyanin. Then this pair of electron is being passed to chlorophyll A. So this is a cyclic process. But in the case of non-cyclic photophosphorylation, the electron never comes back to photosystem 2. That which is lost in the photosystem 2 never comes back to photosystem 2. But in cyclic photophosphorylation, the electron, that is pair of electron that is lost from photosystem 1, is again returned back to photosystem 1. This is a cyclic process, so we called it as cyclic photophosphorylation. In cyclic photophosphorylation, only photosystem 1 is involved, and photolysis of water also does not take place, and only ATP is synthesized, and there is no reducing power, that is NADPH. But in the case of non-cyclic photophosphorylation, both photosystem 2 and photosystem 1 both are involved in non-cyclic photophosphorylation. But in the case of cyclic photophosphorylation, only photosystem 1 is involved, and in that the pair of electron is being passed to chlorophyll A. So we said this as cyclic photophosphorylation. This is all about cyclic and non-cyclic photophosphorylation, which is taking place in the light reaction.